Hi, so let's talk about bacterial genes and genomes with an emphasis on gene transfer, which is something that bacteria actually excel at. So I think the best place to start, this is not new material. This is a review of all those lovely prerequisite courses that you had to take where you learned about the structure and function of DNA and how your genes work. So we're going to look at it again, briefly, a bit of a review, and then also bring in some interesting information that you may not have known about genes and genomes, which is coming from recent research and also because bacteria do everything differently. So let's just start by looking at some of those words. So genome and gene, what exactly are those? So the genome is roughly defined as all of the DNA that can be found inside of a cell. The genome of an organism is the stuff that's hardwired into their cells. So for example, I have a picture of E. coli. Of course I used E. coli, the little rod-shaped bacteria. And spilling out of it is its one and only chromosome, which is made out of DNA. So that's the cell's genome. And in addition to that, E. coli can have one or more additional extrachromosomal pieces of DNA, which we'll talk about later. But that would make up, so all of the genes that are found within this great spilling out mass of DNA would be considered the genome of Escherichia coli. So if you follow that, you take one of these little strands and you blow it up, you magnify it by many, many, many times, beyond the scope of any microscope we have, by the way, you will see that the genome is made out of DNA molecule or molecules. In the case of bacteria, it's usually a limited number of molecules. If you look at the genomes of other organisms, there are significantly more. Like, for example, our cells contain how many different chromosomes? We have 46 chromosomes, 23 pairs of chromosomes, all of which are DNA molecules, gigantic DNA molecules in a molecular sense. So this represents a chromosome. The DNA molecule itself is the chromosome of E. coli, which holds its genome. A gene is a piece of that. And the gene is defined by its sequence, and by sequence I'm referring to the sequence of DNA nucleotides that are found. Remember that there are 4 of those, and the order that they are in that gene is actually what determines the message that gene has to convey. So I'm going to call that gene sequence or DNA sequence, just so that you know what I'm referring to. But notice a chromosome or a DNA molecule will have many genes. So there may be a couple thousand genes. Our genomes have 30,000 genes in them. That's of course excluding the microbiome. And each gene has a function. When it is expressed, and what that refers to is the transcription and translation of that gene into a polypeptide or protein. Then that has the expression of the gene results in some kind of trait, whether it is a trait you can observe directly or if it's one that needs to be looked at in other ways, microscopically, molecularly, biochemically. But regardless, gene expression leads to proteins and peptides, which leads to traits, right? So things that can be detected. So this is just a kind of overview of how that happens. So the sequence of DNA nucleotides in a gene is transcribed by an enzyme called RNA polymerase into a corresponding chain, a single-stranded chain of RNA. And there are differences between DNA, deoxyribonucleic acid, and ribonucleic acid, which I'll point out, that permit this kind of copying to go on. So this is an RNA copy of one of the 2 strands of the DNA in that gene. So this is called the sense, right? This is the sense strand of DNA, which gives you a sense RNA. Ribosomes, shown here as looking like a glove, come along and literally clamp down on the messenger RNA molecule that's produced here and reads it. They translate it. And what they're translating it into is a sequence of amino acids where 3 nucleotides equals 1 amino acid. And then those amino acids are placed in an appropriate order in the protein called the primary structure of the protein. And then that once the amino acids are all in place, the structure of the protein changes. It folds up, becomes globular. I like to use that word, which literally means it looks like it's globby. And then it has its function. So what do proteins do? They function as enzymes. That's a big deal. They're also structural. So, you know, hair, nails, that kind of thing, they're made out of protein. Muscles made out of protein. If you look at us in the big picture. But, you know, in a larger sense, the enzymes that are produced from gene expression are the ones that catalyze the reactions that keep us going and can also result in the development of traits. So that's what gene expression refers to. And gene sequence and gene expression kind of, you know, it's expression of the sequence of nucleotides in that gene that results in the trait for that particular gene, which is also known as a genotype. So let's go look at the structure of DNA first. This should be total review for you. Remember that deoxyribonucleic acid is one of the 4 types of molecules, macromolecules, or what I call biomolecules that make up cells. This one is not directly, you know, DNA and proteins, DNA and RNA and proteins are not so much sources of food and energy, right? They're not carbon and energy sources, although proteins can be, as so much as they have other roles. So the specific role of DNA is to store and transfer all of our genetic information. So the blueprint that makes us us, and by us I'm including all types of cells, not just humans, but all types, is mapped, if you will, into the genes that are found within particular genomes. So remember that a DNA molecule is made up of smaller subunits, right? Those metabolites that I mentioned, which are called nucleotides. And the nucleotide is a complex molecule in and of itself, which has a sugar, right? So it's either ribose or deoxyribose. In DNA, it is deoxyribose. And then you have a phosphate group, and then you have attached one of 4 different bases. So it's either adenine, guanine, thymine, or cytosine. Here's the chemical structures of those, right, shown here in this kind of view. And they are different for a reason. And just to give you a hint, the reason is because the way these molecules are designed allows them to match up with and form base pairs with their matched pair. So adenine pairs with thymine, and you'll notice adenine has this 2-ring molecular structure. Thymine has a single ring, same with guanine and cytosine. And here's the thing, when they line up appropriately, what drives them to come together are, guess, you guessed it, hydrogen bonds. So the hydrogens that are attached here hook up with oxygens or nitrogens on the appropriate bases, but they have to align appropriately or those hydrogen bonds can't form. So first you have chains of nucleotides forming strands, and then the 2 strands come together by base pairing. And then the whole molecule twists up and becomes what's referred to as a double helix, a DNA double helix. So it is 2 strands of nucleotides held together by hydrogen bonds, and then, you know, all brought to you thanks to water, the hydrophobic, hydrophilic interactions that these biomolecules have with water is actually what, you know, now notice the data on this article is, actually it's 2016. So we've known this for a long time that their DNA does form a double helix, but the role of water, the specific role of water, is just becoming more and more known as people start looking into this using biophysics techniques. Okay, so that's our basic overview of DNA structure. Remember, your genome inside the nucleus of your cells are 46 different molecules, each containing millions of nucleotides that hold your genome, just like E. coli has one that has 1 chromosome with 4+ million different nucleotides in it. Okay, so let's look at DNA function. And I have to say that nature did a really good job. Nature always does a very good job. So this molecule was designed to be profoundly stable, which means that genomes can stick around for a really, really long time. As you can tell by our mummy friend here, this was a report that was published in the journal Nature not too long ago, where mummies have been found in all over the world, and they are able to extract DNA from them. And once that DNA is extracted, they can use molecular techniques to determine the DNA sequences. And so in this particular article, they found all these mummies in Egypt, and they were able to trace the origins of those mummies back to their modern day relatives, which was a very interesting paper to read, obviously, since this mummy thinks so. So it's perfectly designed to last for a really long time because it is very stable. And the second thing about it, it is perfectly designed to be copied, which is that back to the hydrogen bonds and base pairing and those principles. And it's so stable that it can come apart to be copied and then go back together again. It's the basis of life, if you want to get, so I don't, not to be too overly dramatic, but it is. So how does that copying, how does this occur? And generally the way of looking at, there's two ways that DNA information can be transferred. One is it's copied into new DNA molecules, and that occurs just before cellular reproduction. So that process is called DNA replication. It's an enzyme catalyzed reaction, which you spent lots of time learning and studying. And then the other is it is transferred into, as I mentioned, traits, and that's through gene expression. So the DNA, the gene sequences are transcribed into RNA, which are translated into proteins, which end up with developing those traits, whatever they happen to be. So DNA is perfectly designed to do its job, and RNA is too. So just to kind of go back and review RNA structure, RNA structure is pretty much the same, except that rather than have a double helix, RNA molecules are single-strand of nucleotides. There's a reason for that, because RNA is not designed to be super duper stable. It's designed to be there for a little while it's needed as an assister, and then it's supposed to go away, because once its job is done, you don't want them hanging around. So it's also designed similar. So here's to compare and contrast the two types of nucleotides, DNA nucleotides. Remember, the sugar is deoxyribose. It's a different sugar, but very close, very similar in molecular structure in RNA, called ribose, right? So this is a 5-carbon sugar, they both are. Ribose has one less oxygen, or excuse me, it has one more oxygen than deoxyribose. And then the other thing is, they both are bonded to phosphates on one side, and on the other side, they are bonded to a nitrogenous base. So the 4 DNA bases, adenine, guanine, cytosine, and thymine. In the case of RNA, thymine does not occur. A base called uracil, a nitrogenous base called uracil, is present. Notice how similar in molecular structure they are. So at this 1 carbon right here, there's a hydrogen, which in this 1 carbon here, there's this group, the methyl group. So they're similar in structure, but that one little difference is what our cellular machinery is able to recognize, making RNA different from DNA. So the sugar is different. This base is a substitute for thymine, right? So they, in DNA it's thymine, and RNA it's uracil, and RNA is a single-stranded molecule. So therefore, just by the nature of it being a single strand, it is less stable than our double helix. And I also want to point out that even single-stranded small RNA molecules can have areas where they fold up to form double-stranded regions. And most of them actually do, with the exception of messenger RNA. The secondary, so-called secondary structure is there to offer it a little bit more stability. You'll see this when you see transfer RNA molecules, which are always shown looking like clover leaves or little trees, because they're folded up in a particular way, because they have that a particular job where they need to be around for a little bit longer. Okay, so that's the structure of RNA. RNA is an assister molecule. So the DNA stores the genetic information. RNA transfers the genetic information. So it transfers it from the DNA off to proteins. So that is a two-step process. The first being transcription, which is when the enzyme RNA polymerase makes a copy of one of the strands, the coding strand of DNA, into a messenger RNA molecule. So it is a one base for one base copy of the DNA sequence, only it is constructed in RNA language. So then that messenger RNA is translated. So it's sort of the interface between, you know, the computer that is the language in our computer and our, you know, interfaces, the Microsoft Word that we use, so that the words that make sense show up to us on the screen, as opposed to the, you know, the stuff that's hardwired into the computer with the ones and the zeros in lots of different random orders that nobody can understand, with the exception of programmers, I think. So that being said, ribosomes will read the messenger RNA because it's written in a language that the ribosomes can understand. And then the reading of that message is what results in the appropriate order of amino acids being made into a chain, and then that chain folds up to become a polypeptide or a protein. So messenger RNA is obviously the go-between. There are other types of RNAs, and the ones you remember, I'm sure, are transfer RNA molecules, which transfer the amino acids from the cell pool to the ribosomes to line them up so that they can be put into place in the protein amino acid chains. And then the ribosomal RNAs are small RNA molecules that are literally part of the structure of the ribosome molecules themselves. And then there are actually other types of RNAs. There's antisense RNAs, there's microRNAs, which all have very interesting roles. And we're just now, I mean, within the past 5 to 10 years, I think, trying to decipher what their actual role is in biology. What are they doing, and how might it be possible for us humans to tap into that information to develop drugs, to develop diagnostic tests? Because again, remember, there's a very specific matching that occurs between genes and RNA molecules. And so, you know, we're trying to tap into that power with research. Okay, so DNA, RNA, this is a representation of something that you might remember as the central dogma, which is the word that Francis Crick of Watson and Crick fame in 1953 said, this is the central dogma of life. So I hope by now you realize I hate the word dogma, because dogma implies that's it, and science is never ever settled, should never be considered settled, and anybody who thinks so doesn't actually know what science is. So this is pretty, you know, we thought it was pretty invariant. This is it, this is the fact. And, you know, it's still pretty close, although we've figured out that life bends the rules. You know, that dogma that we thought we got this figured out. Yeah, so not so much. We're still working on it, and we will be forever, but that's okay. But the central dogma starts with DNA, which is transcribed into RNA, which is translated into protein. And I just want to go back and just say, you know, the central dogma includes the fact that DNA serves as a template to copy itself so that it will be able to be passed to new cells, right? So DNA replication is part of this dogma. DNA is replicated just before cells reproduce. The entire genome is copied. This protein synthesis is from one single gene at a time. And I'm not saying that we only express one gene at a time, because within our genome of our 30,000 genes, many of them are being expressed at the same time. but it is at a gene level that we are talking about it at this point, okay? So DNA transcription is catalyzed by this enzyme, RNA polymerase, forming a messenger RNA molecule. And then also these non-coding RNAs, which I'll just brush over. And then the messenger RNA is translated by the ribosomes into a protein. So proteins are the ones responsible for the traits. But so that is, you know, proteins determine the traits. Enzymes catalyze something like, you know, an enzyme will break down a particular pigment and turn it into a different color, and then that is how your hair ends up a color, that kind of thing. So proteins control traits, which are visible entities, you know, things that we can observe. But interestingly, those same proteins also regulate the expression of genes. So those proteins will go back and impact one of these things to regulate how much of that protein or other proteins are going to be produced. And then they also control cell function on many different levels of that. So it isn't just, you know, what's hardwired into your genes. I want to say that it's part of it, but it's also important to consider that gene expression, what is in those genes, the information in those genes, is regulated or controlled, and it's controlled at many different levels. Shown here are only a few. So you can, you know, by regulation of gene expression, I'm saying you can switch genes on and off, so express them or not express them, depending on what the cell feels is the need for that gene, the expression of that gene. They can be speeded up, speed up expression so that there's more of it produced. You can slow it down. You can change how the cell actually produces something using gene expression methods. So there, you know, notice the three basic levels. You can turn a gene on or off at the level of transcription. So if you prevent the messenger RNA molecule from being made, there won't be any protein. So there's a switch at the beginning of genes where you can turn them on or turn them off at for transcriptional control. At the level of translation and post-translation, there are many different ways that gene expression can be regulated. And again, a lot of which are products of gene expression from other genes will come in and play a role in speeding up the rate, slowing down the rate, you know, changing the mRNA lifespan, for example, making it live longer so you can make more copies from it, that sort of thing. Okay, so I also want to point out that regulation of expression can result in varying traits depending on environmental condition. So I'm going to go to, this isn't E. coli, this is actually a bacteria named Serratia marcescens. It's a gram-negative bacilli. And in the genome of this bacteria is a gene that it has to do with pigment production. So Serratia marcescens is famous because it produces this lovely red pigment, which you can see here. But interestingly enough, Serratia marcescens only produces that red pigment when it is incubated at room temperature. So if you grow that bacteria and leave it in the room temperature incubator, you'll end up with this red, this, you know, kind of cotton candy looking growth on the plate. If you incubate it at body temperature, it produces a non-pigmented phenotype. Same genotype. So the sequence of nucleotides does not change, but at one environmental condition, the pigment is produced, so the pigmented phenotype is produced, and at a different environmental condition, it does not produce that pigment. So it isn't just what's hardwired into DNA. It's also, there's something called epigenetic control. What the environment is telling your genes and how your genes respond to environmental stimuli can result in different phenotypes from the exact same genotype. And there are many, many examples of this, not just in the bacterial world, but I use this example because we get to look at some pretty colored colonies on the plate. Okay, so DNA, what's hardwired in your DNA is obviously important, but there's variable expression of those genes into what proteins are made and which ones aren't. And I also want to add that genomes vary in size. And shown here, so here's, this is the size of the genome, which are measured in BP's base pairs. Okay, so you will also see individual base pairs, 10 to the 11th base pairs. That's a lot of base pairs, right? This is a billion. So this would be 100 billion base pairs in the genome. And you can see that even in the range humans and mammals, right? The sizes of our genomes vary from 10 to the 9th to 10 to the 10th base pairs. Plants, depending on the plant, it could have a few or a lot, right? And then we get down into the single-celled microbes, fungi. This is the yeast. You can see 10 to the 7th. E. coli 10 to the 6th, which of course is the quintessential bacteria model that's always used for these things. Viruses have genomes there, but generally the genomes are small with a limited number of genes, but notice that there are viruses that have very large genomes, such as the Pandora virus, named because it was discovered and it was like, are we opening Pandora's box by playing with this virus? And maybe we are, we'll find out eventually. But the point being, notice the variability in the size of the genomes of all the different organisms. It is large, a good degree of variability. Some have very, very small genomes with only a few genes. Some have pretty large genomes overlapping that of bacterial cells. So genome size varies a lot. And E. coli, as mentioned here, there's E. coli, right? So just this overall structure. So in terms of base pairs, overall, those base pairs can be distributed over multiple DNA molecules, like it for us, for animals and plants, where there's multiple chromosomes. For many bacteria, there are only one or maybe two chromosomes. And then there are also additional extrachromosomal types of DNA molecules that can be found that contribute to the genomes of microorganisms. So this is a list of different types of bacteria, and you can see all the variations in the types of genomes that they have, right? So Agrobacterium, which is a bacteria that lives with plants, has one linear molecule, meaning it is end-to-end this way, as opposed to a circular molecule, compared to Escherichia coli, which has one circular chromosome with 4.5. That's MB, by the way, stands for millibase pairs. So, or KB is kilobase pairs, right? So you can kind of get an idea of the range of sizes for all of these different things. And I always feel inclined to point out that Borrelia, okay, so Borrelia being Borrelia burgdorferi, the cause of Lyme disease, has many other relatives. Notice it has one tiny little chromosome, one linear molecule that's less than a millibase pair, less than 1 million base pairs long. And then it has, when it says multiple circular or linear, there are over 20 different types of DNA molecules that are found within Borrelia burgdorferi. And you will see that these extrachromosomal, this would be the chromosome, the extrachromosomal traits mean that they can pass these things back and forth, that they can express them, they can turn them down. It's being of all the different bacteria that have had their genome sequenced to date, Borrelia is the one that has the greatest diversity in terms of number of chromosomes and different types of molecules that make up its genome. So shout out to Borrelia, which makes it the amazing adapter, the amazing survivor that it actually is. Okay, so genome size varies. Generally speaking, in terms of structure, most of what textbooks will tell you about the structure of prokaryotic genomes has to do with E. coli, because E. coli is always the model for these things. And so when you look inside E. coli, you can see an area called the nucleoid. So remember back to the days when you were looking at prokaryotic cell structure. They don't have a nucleus, meaning they don't have a membrane that separates the nuclear material or the DNA from the cytoplasm, but it is a tightly coiled up, condensed place where within the cell that can take up a half to, almost 3/4 of the interior space in the cell, which is all its DNA. So its DNA is wrapped up tightly and packaged, just like eukaryotic DNA is, but there's no membrane around it. So it is out there in the cytoplasm where the ribosomes are located, where all of the other factors are necessary for transcription, translation, for DNA replication, are present within very close proximity to the DNA itself. So let's go back to E. coli. So we have 4.5 million base pairs. So that's huge. Okay, so on the size of a bacterial cell, I think I mentioned that bacteria are in the range of about a micrometer. That's E. coli in particular. And their genome, their DNA molecule is so big, it's like a millimeter long. Not that you can see it because it's submicroscopic. But nonetheless, if we were able to, and we pulled it out and cut it and measured it, it's 1,000 times bigger than the size of the cell. So what the bacteria do to accommodate that is to fold up their DNA molecules, and they create these so-called looped domains. So that's what you can see here. And they all radiate out of an origin of replication. So this is E. coli again, okay? So there's this origin of replication, which is the site where this all sort of begins. And obviously it's the origin of replication because that's the origin of when DNA starts to be copied. That's where that originates. But you can see the looped domain. The loops are held together by little proteins called binding proteins. And then the DNA molecules themselves, so remember it's already a double helix, but they're super twisted, which is called supercoiling. And that allows it to condense and condense and condense into a smaller thing so that it fits inside a 1 micrometer cell. So we take this 1 millimeter big thing, condense it by supercoiling, tightening it up, flipping it into these little looped domains, and then what you see is the nucleoid. So that's great, but the question is, all right, so we need to express these genes and we need to copy this DNA molecule. So how exactly do we do that? And if you remember, just going back a lecture, that water plays a role in this, okay? But without getting too deeply invested there, here's what I'm talking, here's our looped domains, our supercoiled domains, anchored by these binding proteins. And these something to make note of is these binding proteins are not dissimilar from histone proteins that are found in eukaryotic genomes. And some Archaea actually have histone proteins too, but they're not histones. It's one of those features that makes prokaryotes different from eukaryotes because the prokaryotes have different reasons for producing these types of proteins. So prokaryotes, bacteria, big B bacteria, also have enzymes that their job is to coil and uncoil, so supercoil and unsupercoil the DNA molecule prior to gene expression, prior to DNA replication. Those are called topoisomerases. So they change the topography, if you will. They change the twisted up nature, they untwist it, and then they twist it back up to put it back to storage after it's been used. So one of those enzymes is called DNA gyrase, which your book mentioned. So I just wanted to let you know that it is a topoisomerase involved in the supercoiling process. So all of this, the supercoiling and the genome architecture vastly influence gene expression. So this would be what that looks like in the exponential growth phase. So log phase growth, where the cells are reproducing rapidly, they're expressing genes, so they're opening up this, closing off that, turning on this, turning off that, trying to, optimize their metabolism so they can get as much out of that phase as they can, as much food, as much carbon, as much energy as they can. And then here's the interesting part. So notice when the bacteria hits stationary phase, the supercoiling relaxes. So stationary phase, remember, is a stressful state for bacteria. So they've used up all their stuff. They've reproduced as rapidly as they can. They probably made their environment toxic as all get out. And so now they're sort of like, uh-oh, now what do we do? And one of the things they do is to relax their supercoiling, meaning all of their different gene sites are more accessible, and they will activate certain genes, and mostly they activate almost all their genes, and they do that it's like, oh boy, last ditch effort, what's going to happen to me now? So let's just start expressing genes and hope for the best. And that is how how those topoisomerases actually play a role in gene expression. And we see this in eukaryotic cells as well, because they're, the degree that they are packaged, if you will, how tightly they're packaged is directly related to how that gene is expressed. And we do know that areas of our genome that are, tightly packed up and they never unpack are areas where the genes are really expressed from those areas. So in other words, this supercoiling, this packaging, if you will, which is also sometimes called packing of the genome, meaning making it into its nice little place, is very much involved in regulation of gene expression. That's true for bacteria. That's also true for eukaryotic cells. That's true for our cells. So still comparing and contrasting, the idea that there is coding regions of DNA. Coding is code for gene expression. So coding DNA means genes. These are genes that can be expressed, versus non-coding. Non-coding are just DNA sequences that are not read, are not transcribed and translated into proteins. So notice in a prokaryotic cell, our good friend E. coli, for example, looking at this, so here's a piece of a 50 kilobase pair section, 1,000 base pairs, well, 50 times 1,000, right? 50,000 bases of the E. coli genome compared to 50,000 bases of one of our chromosomes in the human genome. So notice, genes or coding DNA is shown in green, introns are shown in yellow, and these random repeat non-coding areas are shown in purple. Notice E. coli, most of its genome is coding DNA. Most of its genome are genes. And inserted are a few areas of non-coding DNA, which we'll talk about. These are intervening sequences, also sometimes referred to as transposons, that occur. And you'll notice that bacteria don't have introns. These are intervening sequences that are found in eukaryotic genes that are literally, they have to be cut out of RNA transcripts before proteins are made with them. So those are non-coding regions of the DNA. So look at ours, look at our genome. The vast majority is non-coding DNA. So the size of our genome is several orders of magnitudes larger than that of bacteria, but a lot of it is just this non-coding stuff. And we thought for a long time that the non-coding regions were, we didn't know what they were there for. They're not doing anything. They're placeholders. We don't know what all this is about. It's just junk DNA. It's not junk DNA. There's no such thing. Nature and evolution don't allow that to happen. So there's a reason for all this stuff. We just don't exactly know what it is yet, although we're learning as we go. So you'll notice, our 50,000 base pairs. Here's a gene, there's a gene, there's a gene with introns in them. Again, non-coding that get cut out before the gene is translated into a protein. And then all the rest are these small DNA sequences, repeats. They're called repeats. So these are repeats of sequences that get copied over and over and over again and have accumulated within our genomes. And guess what? A lot of them can be traced back to viruses. So there, and that's something that we talked a little bit about previously, but a lot of these sequences have been inserted into our genomes and have been copied over and over again as part of our natural process. I suspect mistakes, but mistakes are always under the influence of natural selection, something to consider when it comes to how prokaryotic genomes differ in overall architecture than do eukaryotic genomes. Speaking of which, I assigned you an article to read about genome architecture. So I hope that you will take the time to read it. It's linked into our schedule for this particular unit. Okay, so structure of genomes and genes and function of genomes and genes, right? Genomes store genetic information that gets transferred to kids, and the genes are expressed to produce proteins and give us traits and characteristics. So what I want to go to from here is the idea that, the transfer of genes occurs widely. So there are two different ways that genetic material or genes, DNA, right, can be transferred. One is through the idea of vertical gene transmission, which is reproduction, right? So genes in the genome are replicated faithfully so that they are exactly the same. And then the cell reproduces and each of the resulting cells ends up with the same set of genes, right? So that's called vertical gene transmission. That's reproductive. So vertical meaning from parent to offspring. And then there's horizontal gene transmission, which is abbreviated HGT. HGT has become huge recently to us. They've been doing it for a long time, but now we're paying attention to it because we realize that bacteria in particular and viruses and are handing around their genes. They hand them out to other interested parties. Usually a bacterial will hand genes to other bacteria. Viruses hand genes to bacteria, or viruses hand genes to whatever types of cells they are infecting. That would include our cells. So it's the transfer of, and I'm just going to call them genetic elements because they are referred to as mobile genetic elements, or MGEs. There's MGE. Mobile genetic elements are, as you might expect, sequences that can move around within genomes, and they can be transferred to other cells, which can then move them around in their genomes. So the recipients of these genes benefit because they acquire new traits, and those traits can be inheritable. So in other words, they get traits and then they hand it off to their kids, and their kids and their kids all have these new traits. And one thing that, as we move into our next unit, one trait in particular that why we have realized all of this in a big way is because one of the traits that bacteria are passing around is the ability to resist antibiotics, which is more than one single gene. It's many different types of genes, and I call that giving them superpowers. So in my little gift down here, this is sort of how HGT occurs, right? So give it out. Here's free money. Here, take this, try it on, see if you like it. If you don't like it, you can pass it on, or you can keep it, or you don't keep it if you don't want to. I don't care. It helped me see if it works for you. And that is the heart, if you will, of horizontal gene transfer among bacteria. So let's do a close up on these MGEs, mobile genetic elements. So they're mobile because they can move around within a genome, right? So like something in the genome of one E. coli could get up and move to another place in that genome, or they can be transferred between cells. And there's several mechanisms that cells can use for that. So this was discovered back in 1931 by a researcher named Barbara McClintock, who was the first to note the idea that there were these jumping genes. And she was also the first to note that gene expression was under epigenetic or above genetic control. And everybody thought she was whacked out because for lots of reasons in the 1930s, there was still very much dogma-driven stuff going on. And it was like, no, DNA, you know, it's solid, it's stable. There's no way that a gene can get up and jump and go over someplace else, because that's what they originally thought of as jumping genes. And now that as sort of the discovery of the first mobile genetic elements, now we know that there are other types. And, you know, just to mention a few here, because I suspect there are more, the one right after transposons, plasmids were discovered. Then it was realized that there are jumping genes, genes that jump up and go to someplace else in the same genome. But then there is this idea of retrotransposons, which are, they don't get up and jump, they are copied, and then the copy moves around in the genome. And then there are bacteriophage. If you remember when we talked about acellular parasites, we talked about viruses that infect bacteria. They are called bacteriophage, right? The bacteria eaters, because that's what was observed first. So bacteriophage are viruses that infect bacteria. And bacteriophage leave elements, let's just leave it at that because you'll see what they are. They can leave elements, genetic elements, in the genomes of the bacteria that they infect, that they can transfer from other bacteria that they have infected previously. So what are these things? Plasmids are extrachromosomal, meaning they are completely separate from the chromosome, as you can see here. So by the way, these two bacteria are two different species of bacillus. You met bacillus as the gram-positive endospore-forming aerobic bacillus. And this one, Bacillus cereus, you actually met in lab, if you remember this one. Cute little guy, you looked at its endospores. But notice that Bacillus cereus has evolved a new species, which is now called Bacillus anthracis, because why? Bacillus cereus acquired two plasmids from external sources that encode toxin genes. So the plasmids are always labeled with a little p, so this is XO1 and XO2 plasmids. On these extra pieces of DNA, which are not the same size as a chromosome, by the way, they're usually very much smaller, just, you know, because the graphic doesn't do it justice. So the chromosome takes up most of the space in the cell. The plasmids are smaller. Two separate plasmids, each carrying genes for two separate toxins, which makes Bacillus anthracis the cause of anthrax, which is, as you know, that probably invokes fear into your hearts, because it's the stuff that people use for bioweapons, because Bacillus is an endospore forming bacteria. So the bacteria themselves, you could disseminate them or spread them through endospore spread, which you can make by just leaving them, you know, treating them poorly, and they will form those endospores. When they reactivate, they will produce those toxin genes. And so that's, you know, we have Robert Koch to thank for the discovery that this particular species was the cause of that disease called anthrax. So those are plasmids, okay? So plasmids carry a few genes. Sometimes they are part of the actual genome and they're sort of stable and permanent. Other times they're transient and they are accepted and then they are lost. The genes are typically not housekeeping genes, meaning they're not essential to the survival of the cell. They produce, like in this case, these toxin genes. So it gives the bacteria superpowers that are helpful to survival for the bacteria, but it doesn't, you know, it does fine even without those genes. It's just under certain circumstances, like for example, when they're in an animal host or elsewhere, they can produce these toxins for whatever evolutionary purpose they use them for. So those are plasmids. Transposons, and again, shout out to Barbara McClintock for discovering these, are DNA sequences that can actually move. So I mentioned that there were two different types of these. There were the DNA transposons and there are the retrotransposons. So again, DNA transposons are jumping genes. They are little sequences that can be clipped out. And notice once they're clipped out, then the two edges here of the DNA, the donor DNA seals up and it goes off to be what it was before. But the transposon inserts itself someplace else. All right, so the recipient, if you will, or the target DNA. So this occurs within a cell. This can all occur within a cell. And then on the other side, the retrotransposons, rather than the DNA jumping out to insert elsewhere, the DNA is copied into an RNA intermediate, like a messenger RNA. But then here's the cool thing. The RNA is reverse transcribed back to DNA. So this enzyme, reverse transcriptase, was a fairly recent discovery in terms of all the different enzymes that play a role in genetics. And it reverse copies RNA into DNA. So let that sink in. That's not supposed to work that way, right? DNA is copied into RNA in regular old biology, but for these, RNA is reverse transcribed into DNA. And then that reverse transcribed intermediate is inserted someplace into another chromosome. So this, by the way, is a trait that has been handed around to viruses that have the ability to use this reverse transcriptase enzyme. So if you kind of remember the Baltimore classification system that we touched on briefly, classes 6 and 7 have this enzyme, have reverse transcriptase enzyme. How they apply it is slightly different. So the question is, who came first, the transposon or the retroviruses that have reverse transcriptase, who gave it to the, so it's kind of one of those chicken and the egg types of puzzles, which we'll never be able to answer probably. Okay, so these DNA intermediates, point being, they start out in one location, they end up in another location within the genome of a cell. And there are ways that these transposons can be passed on to other cells. And that's what we'll talk about next, is how they actually go about doing that, okay? And then number 3 are bacteriophage. So this is what a bacteriophage, or a virus that infects bacteria, looks like. So they infect them with their genome. So this is the phage on the outside of a bacterial cell, and it looks like it's pooping, but it's actually, it's injecting its genome, its DNA, into the interior space of the bacterial cell, which, because it's DNA, remember, the enzymes, the machines that are doing the work do not distinguish between DNA from different hosts. They just will go over there and start to transcribe, translate it. And of course, when that happens, the host is making viral proteins and not host proteins. And so the virus is able to take over the bacterial cell host that way. This is how our viruses, animal viruses, and plant viruses do that too. So the viral genome reconfigures the metabolism of the bacterial cell to produce new viruses. That it reprograms the host cell to be a virus, little virus factory. So how this results in gene transfer is that during this process, as the virus is going through this process, it can pick up genes from the bacterial cell that it previously infected and bring them into the cell that it infects the next time through. So there are a couple different ways this happens. Bacteriophage are actually pretty amazing when it comes to mutations and evolution in terms of reshaping genomes. I think bacteriophage have played a very significant role. I'm not the only one that thinks that, by the way, many people do. So what are these methods of gene transfer? So there are these 3, transformation, transduction, conjugation. So what do these 3 words mean? Transformation occurs when, and these are again in bacterial cells, when you have a donor cell that has all this DNA in it. The donor cell dies, for example, and when that happens, the cell wall breaks down, and then you've got all these pieces of DNA loose in the milieu, right, the environment. And so what will happen is other cells that are nearby will go over and see all this DNA, which looks like junk, right? It's like, look at all that junk. and they'll start reaching out and grabbing the DNA and take it across their cell membrane. And sometimes they actually go about inserting it into their own genomes. Sometimes they just pick up plasmids, right? So it stays extrachromosomally, but other times they'll just go out there and it's like, oh, look, DNA, and grab it. And they literally grab it. I'm going to show you a picture of that in a little bit, I hope. So all that point being, I mean, to me, this seems like unprotected sex, right? It's like, yeah, give me your DNA. No, I would not. No, that's not a good idea. But for a bacterial cell, it is a good idea because why? You know, as an entity, you reproduce asexually. All your cells are the same. If you don't have some kind of genetic variability, if something bad happens, everybody's going to die. So bacteria feel that they can get a few other interesting genes to maybe help them in the event of a catastrophe. So they go out there and they grab that DNA and take it inside their cells. We can now do this artificially. So we can actually take reconstructed DNA molecules and transform bacterial cells with them. This can be done pretty easily in a laboratory. So you'll see many applications of that in terms of we can genetically engineer DNA molecules, plasmids, to hold human genes, and then transform those plasmids into a bacterial cell, which will then reproduce itself and the plasmid, and all the while express the human gene that's in the plasmid. So this is how recombinant proteins are mass produced. Like, for example, human insulin is a recombinant protein. It is produced in bacteria because we've taken the gene and put it into the bacteria using this plasmid approach. So we figured out that that's a good application for it. Bacteria use it for other purposes. So transduction requires bacteriophage. So the process where genes are transferred by bacteriophage is referred to transduction. And then conjugation, talking about unprotected sex, is the production of a structure called a conjugation pilus. Pilus, remember, are those tubes that extend out from the bacterial cell. And in this case, the tube extends out from a donor cell and it hooks up with a recipient cell. And so bacteria are able to directly transfer DNA molecules from one cell to another using this approach. So conjugation is the third overall way. And I just want to point out that transposons can be transmitted pretty much by all three of these methods. So if a transposon is in a bacterial chromosome, it could be transmitted by transformation, it could be transmitted by transduction, it could also be transmitted by conjugation. All right, so let's start with transformation. So in the real world, the bacterial cells go through life cycles and death cycles. When they die, they dump their DNA. Other bacterial cells take them up. Sometimes they will recombine those DNA sequences right into their genomes, and therefore they're acquiring new DNA sequences. That's a form of mutation. And if it turns out to be an advantage to that cell, then it will, so natural selection will select for cells that will be the ones producing that particular gene. Okay, so this is where I was telling you about the fact that this isn't random. All right, so it isn't like, oh, look, accidentally some of the garbage got into my house. This is bacteria use appendages called pili to go out there and grab the DNA. And that's what this is hopefully going to show. Let's just hope that we actually go there and you can watch it. And it's not going to let me, so we'll just exit out of that. Nope, never mind. Okay. Oh, wait, it is going to let me. Okay, so hopefully you are seeing this. So these are bacterial cells. This is called a fimbriae, right? This is a particular type of appendage. And notice what they're doing with it. They extend this thing out and they grab DNA. So the red is DNA, and the bacterial cell is going out there with a claw and grabbing those pieces of DNA. So this isn't random, they go after them. So I found that absolutely totally fascinating, and I hope you do too. I could sit there and watch that all day long, but you know, we need to move on. So go back and watch it if you want to watch them using these pili to grab, harpoon, if you will, and take them into their cells. So in the laboratory, it involves chemical methods. In the real world, they grab them. So, and as I mentioned, we can use that. We can use transformation to transfer human genes, right? Human growth hormone, for example, into plasmids, which we can then transform into E. coli, which will very happily produce a human version of something. Human insulin is produced by bacteria, but it is human insulin. It's not pig insulin. It's not from other humans directly. It's produced, mass produced by bacterial cells that do that very, very happily for us. So that's transformation. I also, in your textbook, talks about this process of transformation from the perspective of the guy that discovered it. This would be Griffiths, one of those. He's not necessarily one of the golden age microbiologists, although he was working during that golden age. He was actually, you know, where you encounter his name, very important guy in microbiology, but he was on that path to discovery of DNA, the DNA double helix, and DNA as the genetic material. Because what he discovered was that bacteria literally transfer physical factors from one to another, which means they can transfer traits, in this case, the capsule, the trait to produce a capsule, which provides superpowers to the bacteria because it prevents them from being phagocytized or eaten. So the bacterial cells are able to transfer the ability to produce a capsule from one cell to others. Okay, so dead bacteria with capsules can transfer the capsule trait to other bacteria. That's what Griffith noticed first, and then he realized it was due to a physical factor because he killed those encapsulated bacteria, and still, when they mixed it with the ones that didn't have the capsule, the unencapsulated bacteria picked up something that gave them the power to produce those capsules. And Griffith proposed that it was a physical substance, and from that we ended up with, oh wow, bacteria transfer genes, and that these traits are based in this molecules that is physical, a physical factor that can be transferred to other bacteria. So Griffith used the bacteria Streptococcus pneumoniae, which at the time was a cause of pneumonia, and only the encapsulated form of Streptococcus pneumoniae is the one that is very pathogenic. So, you know, he was looking at this #1 cause of death during that time frame before antibiotics. It was right up there in the top 3, pneumonia, bacterial pneumonia caused by Streptococcus pneumoniae. Was one of those. So that's what he was working on, and accidentally made one of those discoveries that led us to the 1950s and DNA as a double helix. So that's bacterial transformation. And he came up with that word because the phenotype, the trait, was transformed. So the bacteria without capsules were transformed into bacteria with capsules, and that's where that term comes from. Okay, and then there's bacteriophage. So this is like a whole course. This could be a whole course in molecular genetics talking about bacteriophage and how they contribute to gene transfer. So I'm going to try to give you the 50-cent version of that. So bacteriophage have life cycles, and by the way, there's many, many different types of viruses that infect bacteria. A lot of what we know comes from the bacteriophage that infected, guess who? E. coli. So what we found out using E. coli and the different viruses that can affect them is that there are these bacteriophage that will cause an infection. So they attach, they inject their DNA, and once the DNA is inside of the host cell, so this would be E. coli, you can see the red here is representing the phage DNA. There are two possible routes that the virus will take to ultimately further its life cycle, if you will. Okay, because it's, you know, not trying to debate whether viruses are alive or not, but the point being, some viruses will have this, go through this lytic cycle, which is when, as I mentioned, the genome of the virus is transcribed and translated by the host cell, which produces enzymes which degrade the host DNA. So in other words, the host genes are not going to be used anymore, only viral genes. So viral genes are expressed and transcribed, translated, expressed, which one of those would be a gene for the DNA polymerase that makes new genomes, or RNA polymerase, depending on the virus. And then you end up with new capsid proteins being produced, you know, other structures, and then they assemble or are packaged into the virus head, or in the within the capsid, the genome, the DNA is packaged, and then the host cell releases all of the various bacteriophage off to go start new infections. So this is called the lytic cycle because it results in the death of the host cell, which lyses to release all these, you know, baby viruses, if you want to look at it that way, which then go off to find new hosts and etc. So that's the lytic view. So then there is the lysogenic view. Okay, so lytic is virulent. Lytic is destructive, right? The host cell dies immediately as a result. Once it's used up, its usefulness is to produce new viruses. When that's over with, it's out of there, right? The cells are destroyed. So the lysogenic is also called a temperate life cycle, if you will, because the temperate cycle, it's tempered. So in other words, you can see this can go through many, many, many cycles with the bacterial cell just not being killed, or at least not right away. So here's our phage DNA, which you'll notice becomes integrated into the host cell DNA, where it sits there as a prophage. So the prophage genes may or may not be expressed at this time, right? So they might be expressed at low levels, or they might not be expressed at all. They're off, they're turned off. But notice that the viral DNA sequences are now part of the host cell DNA sequences. And so this prophage is replicated along with the host cell chromosome. So here's our offspring cells with the exact copy of the genome. The prophage is part of the genome. And so this bacterial cells can undergo many, many different replication rounds, hundreds of replication rounds, and, you know, just mass producing bacterial cells which have incorporated in this prophage or this phage DNA. And then that can go on for a long time, but then at some point, the phage is induced. And what that means is the bacterial cell, a switch is flicked to cause that the virus can actually tap into. The signal to the bacterial cell could be something like stress, stationary phase, could be heat, could be lack of food, starvation. Heat and starvation are very stressful things for E. coli in particular. And so the virus says, oh, this host cell is under stress, therefore I need to get out because otherwise I'm going down with the ship. And so they are induced to return to an active phase and enter the lytic cycle and go through this life cycle. So the phage propagates itself at the expense of its host cell. So this prophage state is, when I was talking about bacterial phage elements, that's what I'm referring to. So, and by the way, all of this came from this article. I wanted to just give you the reference for this particular diagram if you wanted to go read further about this article and about all these different types of MGEs that are associated with bacteriophage. So here are 4 of them. Okay, so this actually is the caption to that particular figure that's from that paper. So how do phages contribute to bacterial evolution? So by transduction, by transferring genes among different bacterial cells. So these are the 4 ways. There's others, okay, but we'll just stick to these. Generalized transduction, specialized transduction. Generalized transduction occur when in that lytic cycle, the bacterial cells, their DNA is ruptured, broken up into small pieces by the virus. And then when the virus is packaging up its own DNA into its head, into its capsid, sometimes it grabs some host DNA also. And so the virus can, by virtue of almost a mistake of just the packaging process, can pick up these random pieces of either the host DNA, the host bacterial cell, or plasmid DNA, and then transfer them to the next bacterial cells that they infect. So that's generalized transduction. So specialized transduction is when a prophage cuts itself, it goes back into the induction phase, and as it does that, it excises or cuts out part of the host cell chromosome as well. So you're transferring not random pieces, but pieces that are next to the prophage to new bacterial cells. And then phage conversion occurs when a prophage, as I showed you, carries a gene that when it is in the host cell, that gene is expressed. So you will see that many bacteria that started off in life as not necessarily being all that toxic in terms of disease-causing abilities have acquired through this process of phage conversion genes for toxins or genes for other virulence factors that cause them to cause diseases. So, just looking at one bacteria, I mean, there's lots of examples of this, but bacteria that, Streptococcus pneumoniae, it had genes for, capsule or not capsule. The capsulated one made it very dangerous to us. The unencapsulated version is very low danger to us. So capsule is one example, but I'm talking about usually toxin type genes that are found on prophage molecule, the prophage, the virus itself, that are expressed when the virus is in that prophage state. So that's what phage conversion refers to. And then sort of scary to me are the newest of these discoveries, the gene transfer agents, or GTAs, are prophages that got, kind of degraded, and so they can't actually go back into their, induction state. They can't actually go back into active state, but they're nonetheless carry these genes, and the genes for toxins, again, could be transferred because they're part of these prophage elements. So there's a lot that has to do with antibiotic resistance and the transfer of antibiotic resistance genes. And again, the role of bacteriophage in the HGT related to the antibiotic resistance, with the rapid rise of antibiotic resistant bacteria in particular, can be traced a lot to these bacteriophages. So stay tuned for that. Beyond this course, you'll probably hear more, because those of you that are going into medicine, this is a significant problem. Okay, so I took that figure and I redid it with where those, the different types of HGT that can occur as a result of transduction occur, right? So notice in the lytic phase, generalized transduction occurs at the stage of DNA packaging, where the little bits of host chromosome get picked up and stuffed into the capsid head, which then, as those new phages are released and they go through this, they can transfer that DNA to new infected host cells. So on this side, notice phage conversion occurs during this prophage period, where if this prophage, if the bacteriophage itself has genes for toxins, while it is part of the prophage, those toxins can be expressed, okay, and reproduced and transferred to others. So that's what phage conversion refers to. And again, here's this, the idea, or the prophage started off this way, but after many, many rounds of reproduction, something happened to it, it's degraded, not really working out all that well. Domestication, if you will. I'm sure evolution is involved with this, meaning, oh, I like it better in this cell. I don't really feel like going out and having to make a living as a virus. I'm going to stay over here. And those are where these GTAs come from, which carry genes that can, again, give bacteria superpowers. And then specialized transduction occurs at this stage, where the prophage is excised, or the genome of the virus extracts itself from the host cell to launch it into the lytic cycle again. So obviously, bacteriophage have a very complicated relationship with the cells that they infect. And I want to say there are viruses that infect all types of cells. And we all have this relationship with viruses. So viruses that do this, they do it to make a living. They're the ones that are taking over the genetics of the cells that they infect, if you will. And over time, which you'll be hearing about as we move into Unit 4, more and more about how we evolve with these viruses in terms of, usually a virus starts off in a dangerous state, so it makes us very, very sick at first. But then over time, it's like, well, maybe that didn't work out so well because I ran out of cells to infect. So I need to temper it back down. And this is a give and take that has occurred since probably the beginning of history. And by that, I mean the history of life. And we'll just put it life on Earth because that makes it easier. Okay, so takeaway is, mostly your book focuses on generalized and specialized processes of transduction, just so that you know that. And then conjugation. And again, this is, there are genes on plasmids that tell the host cell to produce this protein called pillin, which results in the formation of a conjugation pilus, a tube. Okay, so that the pilus that I showed you going out there for the cells to go grab the DNA for when they were busy doing transformation is also made out of this type of stuff. These are tube-like structures that go out. In the case of grabbing up DNA for transformation, that's the job, is to just go suck it, be a suction cup. In the case of conjugation pilus, the pilus is extended to another cell, enters that other cell, and now you have a passageway between cells where one cell, the donor cell, can actually donate DNA from one cell to another. So usually the donated DNA is the plasmid itself, the plasmid that carries the gene that allows them to make the pilus are called F plasmids. F stands for fertility, as from back in the day, right? So this would be an F plus donor cell has a plasmid, which is an F plasmid, meaning it has the fertility gene, meaning it can make this conjugation pilus. This is the recipient cell. So you'll notice what happens. The F plasmid gene is expressed, the pilus is produced, and then the plasmid is copied so that it will be transported over to the other cell where you end up with an identical copy in the second cell. So donor cell, recipient cell. So you go from, notice the recipient cell starts off without the plasmid. By the time it's over, it has the plasmid. So conjugation is a way for bacteria to directly transfer genes from themselves over to others. And again, shown here is just the transfer of the plasmid. Many plasmids carry genes for antibiotic resistance, for example, carries genes for toxins, carry other types of genes that are very useful to bacteria. So they're happy to accept that little piece from a donor cell. And then, sometimes as opposed to the plasmid staying as a separate extrachromosomal bit of DNA. Sometimes plasmids can actually integrate or become part of the host cell genome. Similar to what you saw with those prophages, right? So they're plasmids that can also recombine or insert themselves, if you will. And then you'll notice that you know, the integrated plasmid can transfer some of the host cell genes, not just plasmid genes, but host cell genes during that process of conjugation as well. So the point being, bacteria have all these ways that are known to transfer genes amongst themselves. And sometimes it's very specific. So, you know, it's only like from an E. coli to an E. coli. Other times it's just, you know, I'll take genes from anybody. So it can be quite a random process. And the viruses, the bacteriophage, obviously play a very major role in the evolution of bacterial genomes. So when we look at evolution of bacterial genomes, realize that bacteria excel at acquiring new traits. Sometimes they're superpowers. And by superpowers, I mean that makes it so that we can't kill them. They can make us very sick, or other types of superpowers that they might acquire. And it also leads to evolution of new species. So it's not just new traits, but it's completely different branches of life. So obviously this has been happening since the beginning of time, since there was the first cells were produced, and once they had packaged that nucleic acid into a cell membrane, you know, these processes probably began. And, you know, the question again is the role of viruses. Were the viruses here first and started that process? Did the viruses pop out of the genomes of cells as they were being developed? Well, you know, I don't know that we'll ever be able to figure that out, but biologists love to argue about it. So I don't know that I think humans are the only creatures on Earth that stand around, you know, by the coffee machine talking about whether any of this matters. Because, you know, it's like, well, that's what it is, and not a ton that we can do about it, but there you go. So notice evolution of new traits through mutation, deletion, right? Mutations, meaning the removal of bacterial sequences or DNA sequences, removal of genes. They can acquire genes by HTT. They can rearrange genes that can result in new and interesting genotypes and phenotypes. But notice, at the end of the day, it's natural selection that plays a role in deciding which of those genes are going to stick around long enough to participate in the evolution of new species, right? So over time, from a common bacterial ancestor, the intracellular parasites, the ones who decided the best thing for them would be to make a living by growing inside of other cells or inside of other organisms, they didn't need all those genes. They're like, I don't need this, I don't need that. So they reduced their genome size so that they have the appropriate genes that allow them to survive in a host. Those that live extracellularly, they're the ones who are like, give me the genes, give me all the genes, because the real world sucks, it's hard, and give me all the genes that you can give me, and I'm going to try them all out and see which ones work best. And some of them I might keep till later because I might need them later. And then, you know, just realize that mutations occur all the time in all types of cells. And those mutations can stick or not stick depending on natural selection and other, obviously, other types of factors as well. So that gives us, if you want to look at it this way, what I'm showing you here gave rise to that lovely sprouting tree of life, or the shrubbery of life, as I like to think of it, in terms of how we went from, you know, those early nucleic acid things surrounded in a membrane to the diversity, remarkable diversity of life that we have on the planet at this moment. Okay, so where I'm going with all this is that we're moving into the realm of the bad guys, the ones that cause disease. And one of the, you know, unfolding dramas of our time is this expanding, if you want to call it an epidemic of antibiotic resistance among bacteria, where, you know, we very cavalierly thought we had antibiotics, we'll have them at our disposals forever, bacterial infections, no big deal, we got this. Yeah, that was very short-sighted of us. So these very well done frontline episodes. One is called Hunting the Nightmare Bacteria. The other is about the problem with antibiotics, which show all of the different factors that go into the evolution of certain types of diseases. And in this case, these are superbugs. They're called superbugs classically, which are bacteria that can cause infections that are untreatable by any type of antibiotic. And they are able to overwhelm our immune defenses. And it's how we got to the point where we have these things on this planet is truly a remarkable story. So watch these two episodes, watch and take notes, because they do discuss specific types of bacteria, specific bacterial species, and the types of genes that have accumulated to result in this, you know, perfect storm, if you will, of multi-drug resistant microbes. And in the case of some, absolutely 100% resistance, meaning there is no treatment for some of these bacterial species. So that's for you to do next week. And I hope that you will enjoy them as much as I do. There are sick people involved, and I'll just tell you, don't get too deeply invested in any of them as you're watching this story. Okay, so onward to our next topic, and I'll see you next week.